Good day, and welcome to CureShank's Resources for Researchers webinar series. This series is sponsored by the CureShank Biomarkers and Outcome Measures Consortium. And I'm Trish Davidson, and I help administer the CureShank Consortium. One of CureShank's goals is to help grow a community of people who share a common passion for improving the lives of people with shankopathies. And CureShank wants to enable collaboration and productive relationships among all of the stakeholders in our field. Today, we will hear from AutismBrainNet about their resources. And our hope is that making connections and introducing you to these resources will enable all of us to work with speed and efficiency to develop treatments for shankopathies. And we are pleased to have Dr. David Amaral, Scientific Director for AutismBrainNet, and Carolyn Hare, Clinical Director for AutismBrainNet, with us today, who will share more about the resources that are available to you. Next slide. And we have an exciting agenda for you today, and I'll serve as moderator for this webinar. As you look over today's agenda, we have just a few housekeeping notes. Following an overview of CureShank, you'll hear a presentation from Dr. Amaral and Carolyn Hare from AutismBrainNet. The presentation should last approximately 30 minutes, and you're welcome to submit questions through the Q&A feature during the presentation. Participants will be muted, and your questions will go to our speaker panel at the end of the presentation. Our speakers will answer as many questions as possible. And we also will have an email available for any questions that are not able to be answered during the webinar, or if you think of questions following the webinar and would like to reach out for further information. But before we hear from AutismBrainNet, it is my distinct pleasure to introduce Geraldine Bliss, co-founder and CEO of CureShank, who will say a few words of welcome and talk about the work of CureShank. Geraldine.
Thanks, Trish. Talia, can you advance to the next slide, please? Thank you. I'm just going to go very quickly and give a little background on Phelan-McDermid syndrome. Phelan-McDermid syndrome is an autism-associated condition. It's a neurodevelopmental disorder strongly associated with intellectual disability, very frequently with autism, and in a subset of patients, about 40% develop epilepsy. It is caused by haploinsufficiency of the SHANK3 gene. In terms of its kind of prevalence, it really represents approximately 1% of all cases of autism. If you look at patients that have both intellectual disability and autism, it rises to about 2%. The incidence is thought to be at least 1 in 30,000 in Europe, but that's thought to be an underestimate just because of the lack of frequent genetic testing. Could potentially be as high as 1 in 8,000 to 1 in 15,000. And many companies use a figure in the range of 28,000 to 50,000 when they're talking about the PMS market. So that makes Phelan-McDermid syndrome fairly comparable in size to spinal muscular atrophy, Rett syndrome, and Dravet syndrome. Let's go to the next slide. Yeah, here's a little bit about CureShank. It is the mission of CureShank to accelerate the development of treatments for Phelan-McDermid syndrome. And we do that through a variety of strategies. One is just funding PMS research in areas that we identify as sort of areas of great kind of deficit or where there's a need for our funding. And an example of that are 2 recent epilepsy grants to stimulate more epilepsy research in Phelan-McDermid syndrome. One of our biggest initiatives is the CureShank Biomarkers and Outcome Measures Consortium. This is a pre-competitive consortium for industry. It's composed of companies that are either working on SHANK3 or interested to really identify and fund projects of mutual benefit. Our focus here is primarily on biomarkers and outcome measures, but many companies are trying to solve the same problems in terms of how do we conduct successful clinical trials, and the consortium is a way that we can work on this all together. We held a patient-focused drug development meeting a couple of years ago, and it's really important to us that we elevate the voice and the perspective of patients and their caregivers in the whole research enterprise. Let's go to the next slide. A little bit about the consortium, and then Trish, should I hand it over to you after this? Okay. We facilitate a cooperative, globally aligned effort that's focused on identifying and developing novel approaches that can enable successful clinical trials in PMS. This consortium establishes just a pre-competitive space for companies to advance biomarker research efforts and outcome measures in Phelan-McDermid syndrome. And this is open to any company that's interested in Phelan-McDermid syndrome or interested in neurodevelopmental disorders. And if you're interested in learning more, please reach out to Trish. Her email address is provided here at the bottom of the slide. Trish, do I need to tell people more about the consortium?
Well, thank you, Geraldine. I really appreciate it. Just one more thing about the consortium. One of the most essential functions of the consortium is to be able to provide that forum and process for discussion and guidance and feedback among participants. And so please reach out to us if you're interested in learning more. And without further ado, we welcome our speakers that are here with us today. We're honored to welcome two speakers from AutismBrainNet. Dr. David Amaral is Scientific Director for AutismBrainNet, and Carolyn Hare is the Clinical Director of AutismBrainNet. And I'd like to give you just a little background on both speakers before we begin. Dr. Amaral graduated from Northwestern University with a degree in psychology. He then received a joint PhD in neuroscience and psychology from the University of Rochester. He conducted postdoctoral research at Washington University, and he was at the Salk Institute for Biological Studies in La Jolla for 13 years before joining the University of California, Davis as a professor in the Department of Psychiatry and Behavioral Sciences in the Center for Neuroscience. And Dr. Amaral was the Benito Foundation Chair and founding research director of the Mind Institute from 1998 until 2018. Dr. Amaral started the Autism Human Project in 2006 with the collaboration of numerous Mind Institute faculty and staff. And the project has become the largest single-site longitudinal analysis of young autistic children. In 2015, Dr. Amaral became the editor-in-chief of Autism Research, the official journal of NSAR. He also serves as the director of AutismBrainNet, funded by the Simmons Foundation to develop post-mortem brain repository to enhance autism research. He has published over 350 research articles that have been cited more than 83,000 times. He has co-edited 3 books. And in 2019, Dr. Amaral was elected to the prestigious U.S. National Academy of Medicine. Dr. Carolyn Hare will be presenting along with Dr. Amaral. Carolyn serves as AutismBrainNet's clinical director. In this role, Carolyn works directly with donor families to facilitate the donation process and conduct essential clinical interviews. Carolyn is a former special education teacher, certified in ADIR trainer, and research clinician who has dedicated her career of nearly 30 years to learning from neurodiverse communities. Prior to working with AutismBrainNet and its legacy Autism Tissue Program, Carolyn founded Achieving in Higher Education with Autism and Developmental Disabilities, as well as the Arts for Autism Foundation of Pittsburgh. We are delighted to have our 2 distinguished speakers today. Dr. Amaral, would you like to begin?
Thank you, Trish, and it's wonderful to be here. And before I get started talking about AutismBrainNet, I wanted to say that over the weekend, I was reviewing the CureShank website. And I just wanted to say how impressed I am with Geraldine and the whole CureShank team with how much you've done since 2019. I mean, in a very short time, you've established academic and corporate partnerships. You've started, built your conceptual model of Phelan-McDermid. You've started a funding program in a very, very short time. And just to say that we're delighted to be able to contribute in some small way to your research mission and look forward to, a long-term partnership with CureShank.
Thank you so much for those kind words. Can I just say we were so fortunate to be able to build on a really outstanding foundation of science when we started in 2019. And that's thanks to you and many other scientists who have been kind of laying the groundwork for the treatments that are now beginning to happen. So thank you.
You're very welcome. All right, well, let's go to the next slide, which gives an outline of our presentation today. So I'll give a short overview of the organization of AutismBrainNet. Carolyn will talk about the inclusion and exclusion criteria for donations to the network. I'll talk a bit then about the tissue processing and also the current status of our repository, how many donations we have. Carolyn will then go into more detail about the donation process and particularly how she and her colleagues make it easy for families to be able to make a donation in a very difficult time. And then some of the clinical data that she collects, a little bit about how researchers can request tissue from AutismBrainNet through the Simons Foundation, and then we'll be happy to answer any questions that may come up. So if we can go to the next slide. So AutismBrainNet is actually celebrating its 10th anniversary this year. We were founded in 2014. It's a program of the Simons Foundation that provides all financial support and also much of the guidance for the program. Our goal is to build an essential resource for research into the causes of autism and related neurodevelopmental disorders. So really the impetus for this was in part my own attempts before 2014 to do research on post-mortem brains and realizing there just wasn't an adequate number of cases available to researchers to do state-of-the-art research. And so we went to the Simons Foundation initially, we went also to Autism Speaks, that was an initial co-funder. But to indicate that if the state-of-the-art research is going to be done, really you need to have the resource in order to be able to look at postmortem brains. And again, the premise is that to understand a neurological condition or disorder such as Phelan-McDermid or autism more generally, it's essential to be able to look at the brains of individuals that had the disorder and condition during life. I'm going to go into more detail about this in a bit, but since 2014, we've been able to collect 388 postmortem brain donations, and we're increasingly distributing that tissue to researchers worldwide. So next slide, please. AutismBrainNet is really a network. We have a number of individuals across the United States and actually internationally who are contributing to the effort. We have 3 collection sites, one at the Mind Institute at UC Davis in Sacramento. We have one at UT Southwestern in Dallas, and we have a third at Mass General Hospital in Boston, one of the Harvard hospitals. You can see the individuals that are participating at each of these, we call them nodes, where brains can be delivered. And the idea was that if we have collection sites throughout the country, we could have the shortest possible post-mortem interval between death and when the brain is prepared for science, which is beneficial to doing state-of-the-art research. We also have a number of administrative staff who are helping us. I want to particularly point out Dr. Marta Benedetti, who is co-director of AutismBrainNet and is at the Simons Foundation in New York. I did mention that we have a couple of international nodes. In particular, we have a new node that just has started recently in Montreal at the Douglas Bell Canada Brain Bank, run by Dr. Naguib Mekawar. So regardless of where a donation comes into this network, it's processed in exactly the same way. We have a lot of quality control procedures put into place. And then when an investigator requests tissue, it's sort of like an Amazon kind of thing where the tissue request comes in. We ensure, of course, that the science that is going to be done is going to be done well. But then in order to distribute the tissue, we have a tissue manager who is then able to, and she's pictured at the UT Southwestern node, Kelly Gleason. She can then select tissue from each of the nodes and have it shipped to the investigator, and all this sort of a transparent process. They get shipments of tissue which they can use for their research. Next slide, please. Carolyn?
Thank you. And before I begin, I just wanted to echo David's impressions of Cure-Shank and Geraldine and all of the wonderful work that's gone on. We're really privileged to be here with you today. So in addition to the team that David just shared with you, we're a small but mighty group. And as a result, we find ourselves relying actually quite heavily on our community partners to understand and appreciate the importance of postmortem brain donation for research into neurodevelopmental conditions. We also want our community partners to be quite familiar with the donation process that we'll share with you later so that you can educate folks when the time comes, so that we are continuing to build this important resource for your communities. We hope that in return for our reliance, our heavy reliance on you as community partners, that you also find the service that we provide to be compassionate and important to your communities. Next slide. So in terms of our inclusion criteria, so who can be a donor to AutismBrainNet? Concisely stated, anyone with a diagnosis of autism at any age can become a donor to our program. For our folks that have a genetic condition, as long as it is related to autism or part of the SPARC gene list, obviously including SHANK3, Phelan-McDermid syndrome, other related neurodevelopmental conditions, you also are an important potential contributor to AutismBrainNet, even if you do not have a diagnosis of autism. And then for our, I think a lesser known part of our initiative is finding important comparable control or typically developing matches for our donors with a diagnosis. Those folks, we're looking to build our cohorts of folks under the age of 50. So again, this is for people without a diagnosis, up to age of 50. No known confounding diagnoses, so no neurologic, serious psychiatric, no acute or chronic brain injuries. We do include also adolescents and young adults with a sudden unexplained death. Next slide, please. I jumped a little bit ahead to our exclusion criteria, but for our control donors or our donors without a diagnosis, there are some exclusions. It would be for folks that have an acute or chronic brain injury, any prolonged chemotherapy that can affect the quality of the brain tissue, as well as those psychiatric and neurologic diagnoses that I was just talking about. We do also screen for folks with known bloodborne pathogens. That would be a rule out to be a control donor to our program, as well as chronic, and this is significant long-term alcohol or substance abuse that might compromise the brain for research purposes. Next slide.
Okay, so I will then say a little bit more about our collection. I already mentioned that we have 388 donations. If we can go to the next slide, please. So this is a breakdown of the components of our collection at this point. So we have 155 cases of individuals that had a diagnosis of autism. This is both autism, where Carolyn and her colleagues have confirmed the diagnosis. So one thing that's important is that we, once a donation is made, Carolyn works with the family to carry out questionnaires to ensure that the individual actually had an autism diagnosis. So the 155, the vast majority of them have already been confirmed. There's a small number, I believe it's 16 or so, that are still in the process of being confirmed. You can see in the different shade of green down below, we have 4.12% of individuals that we call ASD-NC, non-confirmed. So it is the case that some donors come to us, they never had a formal diagnosis, but the strong indications that they did have autism during life. But Carolyn is not able to confirm for a variety of reasons. And we feel that this is important because autism is already heterogeneous enough. And so if scientists are going to do studies, they want to ensure that at least they're starting from the vantage point that the donor tissue came from an individual that actually had autism, and doesn't want to be increase the variability by also including individuals who may not have had autism or may have had some other disorder that's not related. So that's why we go to the extra step of ensuring that all individuals have a diagnosis of autism, if that's what they come in as. You'll see that there's also a large category, NDD. This is neurodevelopmental disorders without a diagnosis of autism. This is where Phelan-McDermid without an autism diagnosis would fit in. And there are 46 of those. We are doing whole genome sequencing on every brain that comes into AutismBrainNet at this point in time. So even those individuals who may not have had a genetic diagnosis for their autism may ultimately have a genetic condition ascribed to them through this post-mortem analysis. Next slide, please. So just for the researchers who might be listening, when the brain comes into one of our nodes, they're all prepared in exactly the same way nowadays. Half of the brain is frozen, is sectioned into coronal sections, about 12 millimeters, and then rapidly frozen. And that tissue is generally used for genetic studies of one kind or another. The other half of the brain is fixed in aldehydes, which is typically done for more pathological studies or histological studies. We've adopted a procedure where we just minimally fix the tissue, which makes the tissue optimally beneficial for all kinds of immunohistochemical studies or histochemical studies or pathological studies. So that's the way we currently process the tissue. We've evolved over the 10 years of our existence. When we first started, brains were, half of them were frozen, half of them were long-term fixed, which is sort of more the standard for brain banks. We've stopped that as of almost 3 years ago now, and use this minimally fixed process, which we think is going to be better long-term. Once the brains are processed at a node, they can then be valuable for decades. So they go into cold storage. And what we know is that tissue can be used for research, literally for decades and decades. And I wanted to point out that it's the goal of AutismBrainNet to make each case available to as many researchers as possible. So again, in the old days, a brain would come in and portions of it would be allocated to a particular laboratory, and that would mean that no other laboratory would get tissue from that part of the brain of that individual. We've actually adopted procedures where we can provide tissue from each brain to tens, if not hundreds, of researchers, and again, over many, many years. So we're trying to optimize this precious resource as a valuable resource for research. Next slide. I'm not going to go over these numbers in detail. If a researcher is interested, I should say that we have a very extensive database of all the information that we get about each donor and on every aspect of how the tissue is processed. So when an investigator comes to us and says, I'd like a certain number of cases, with these characteristics, we can go into our database and find them. And this slide simply says that we have both tissue, we have, for example, tissue from 100 cases that tissue of autistic individuals from 100 cases that was rapidly frozen, from a few more 105 cases that was fixed. So we have tissue to support all kinds of research. And Kelly Gleason, who I mentioned previously, works with the investigator to ensure that they can get the tissue that they need to optimize their specific research questions. Next slide. This is one of the things that's important to know is that the length of time between when an individual passes away and the donation is, or the brain is processed, that's called the post-mortem interval, is critical. The shorter that is, the better the tissue is for doing research. As you can imagine, the brain, like any other organ of the body, once a person dies, it starts to change. And those changes then don't reflect how the brain was during the life of the individual. So we want to try and capture the brain as it was in life. And so we want to try and minimize that post-mortem interval. What this shows you is that, we have a criteria that we try and get all brains that come in prior to 48 hours after death. Really, what's optimal is within 24 hours after death, or even shorter, if possible. And for that reason, and Carolyn will say more about this in a future slide, it really is important for families to talk about the idea of donation early, to have a notion of whether this is an option they want to exercise, and to discuss it long before the need ever arises. Because if you can make that call to AutismBrainNet quickly and get Carolyn and her team working with you, that's going to facilitate a more rapid post-mortem interval and a more optimal donation for research. Next slide, please. we have, obviously, autism has a preference for males in the general population of autistic community. And we have collected, as you can see here, donations from more males than females, although we have a very significant number of females and always, of course, trying to increase the numbers of both males and females that come in. And as you can see, we sort of have a balanced set of non-autistic controls. You can see that we have 120 males with autism. We have 100 controls, and we're consciously trying to accept control donations that are as closely as possible matched to our autism and neurodevelopmental disorders donors, because those are going to be the best comparisons for researchers to carry out their research. Next slide. This is information perhaps most germane to CureShank. At this point in time, we have donations from 7 individuals who had Phelan-McDermid syndrome. There are 4 females and 3 males. The age range for the donors was between 6 and 52 years. And again, just like every other type of donation, some of this tissue has been frozen, some of it has been fixed. And so these cases can support then both genetic or pathological immunohistochemical studies. I think it is possible that there may be more Phelan-McDermid syndrome cases in our collection. We are doing whole genome sequencing, as I mentioned, and for many of our older donors who never had any genetic testing, they have symptoms that are consistent with Phelan-McDermid, and once we identify that they had SHANK3 mutations, that we will be able then to add them to the list. That whole genome sequencing is ongoing. Actually, the sequencing has already been carried out. Right now, we're in the analysis stage, and we have academic colleagues who are helping us with doing the calls for any variants that we might see in the genomes from those donors, and those that sequencing is being done from tissue that is from the brain. We're actually doing it from a portion of the frontal lobe. So and I should say that at the moment we are collecting blood samples when possible from the donors. We are collecting a dermal biopsy. Nowadays, this has been started over the last about year and a half, I believe. And the dermal biopsy is to produce fibroblasts so that we can ultimately expand those and induce pluripotent stem cells. And that's something that, again, if researchers are interested in obtaining, we can certainly discuss that. And the Simons Foundation has a much larger program on induced pluripotent stem cells that researchers can interact with. Next slide. I think we missed a slide, but that's okay. I think it was in the most recent iteration, but it was a slide. Yeah, so if we can just go back for a second. Go ahead and go back one more. There was one more slide that we put in at the last minute, and I'm sorry, but it's no big deal. It's just the age range of donors for AutismBrainNet, and what it shows is that that at this point we're still avidly attempting to collect donations because our age ranges are very patchy, so that there are large numbers of donations in the sort of 20 or 30, 40, 50-year-old range. Not so many younger than that. And even for comparison control tissue, we have very few that are in the very youngest range. So it's an ongoing process of collecting donations. And even though 388 sounds like a large number, really to sort of fill all the gaps in both the ages and sexes of the donations, we still have a long way to go. I do want to say, and let's just go backward one more step, and then Carolyn will start. I do want to say that, sometimes I hear myself, giving all these statistics and and talking about donations and how they're fixed and whatever, and it sounds to me sort of cold. And I want to just emphasize that, it's a real with mixed emotions that we get every new donation, because we realize that while it benefits the repository and it benefits research, it's at the cost of some family suffering a tragedy. And for that reason, I'm delighted that Carolyn and her colleagues really treat the family with compassion and if anything, make the donation process a very positive experience. So I want to turn it over now to Carolyn, who can talk more about the donation experience.
Carolyn, before you say anything, I just want to say I've spoken with several families who have participated in donation, and they just have had so many wonderful things to say about the experience, Carolyn, especially, working with you and your team. Everybody's been so complimentary about the sensitivity. I just want to read a little quote from one of our parents. "I have been blown away by the sensitivity and care that the ABN has had during my brief interactions with them." And I just, thank you so much for making this, making a difficult time an easier process for our families. Thanks to you, Carolyn.
Well, you're very welcome, and I honestly am often asked by friends in particular, and of course colleagues who like you understand that this is a really complicated situation. We want to appreciate or express our appreciation for families making really the ultimate sacrifice, in a moment that is really difficult. We honor that, and from my perspective, it is truly a privilege to be positioned to provide this essential support to our donor families. They are cared for and appreciated in a way that is, as unique as the decision that they've made to donate. So thank you for the kind words, and David, thank you. Appreciate that. So in terms of the donation process, just to be really clear and direct about how this goes, it is very important for families to understand that brain donation is a distinctly separate process and requires proactive action on the part of the family or someone very close to them. It is essential that they call our 24/7 helpline. The number you see there on your screen, 877-333-0999. And the reason that it's important to call that number and specifically to indicate to our answering service that you're calling regarding a death and donation is because that phone call alerts our clinical team that this is an important phone call to answer. And so we do answer that line, as it says, 24/7. At any point, anyone can reach out to us. We will respond. We will walk a family through the informed consent process for postmortem donation. And at that time, we'll also collect the information that is essential for us to begin coordinating the logistics of the recovery to make sure that that donation, as David was discussing earlier, is recovered as promptly as possible and makes its way safely to our closest or on-call repository, where it is then preserved, as David was discussing, in perpetuity to support research for decades to come. The last aspect of this that David also touched on is that we do follow up with every single one of our families when possible. There are times when a family needs some space. We respect that. But when possible, we follow up with our families so that we can learn all about their person. And we want to approach this in a way that feels very conversive, and supportive, very importantly. All the while, we are collecting information that researchers have told us is essential for them to be able to do their work. Next slide, please. So in that conversation that I was just discussing, there's quite a lot of information that my clinical counterpart, Nicole Coman, who is just wonderful,
Oops, did we lose Carolyn?
earlier, the diagnosis that was given during life. Oh.
Carolyn, you blipped out there for a little bit, so maybe go back just a little bit and start with the clinical information that gets acquired.
Sure. And I turned off my camera in hopes that this makes me a little bit more clear. Is this better?
Yep.
Okay, great. So in the conversation that I was describing that we have with families after the donation process occurs, we aim to be most importantly supportive of them and honor their person and take the time to learn everything that there is to be learned about that person as much as we can. The idea being that we're collecting information that researchers, again, have told us that is essential for them to carry out their work. So that information includes the more than 100 questions we ask as part of the autism diagnostic interview. Whether or not a person has autism, we administer this interview typically because it does collect information about early development, how a person interacts with the world around them, any specific interests, sensitivities. Really, it's quite a comprehensive questionnaire. But importantly, in addition to this, we, as David was describing, conduct genomic characterization. We have also implemented what we're calling a research neuropathology report. So those, David, actually, could you describe the research neuropathology a little bit more?
Sure. So for each one of our nodes, the MGH node is directed by 2 board certified neuropathologists. And we've asked them to sample each brain that comes into AutismBrainNet. There's a small number of sections from different regions of the brain, and they can tell us if there's any indication, both from cases as well as from controls, if there's any ongoing problem with the case, for example, vascular changes that might be due to hypertension, things that a researcher would want to know when they select that brain tissue. And again, it's done on every case. For cases that come in through medical examiners or from medical examiners or coroners, excuse me, who request that a full neuropathology report be carried out, then our board certified neuropathologists will carry out a standard neuropathology assessment, write a report just like any other neuropathology report, and that's provided back to the medical examiner for free. So that's a service that we can provide to them. But what Carolyn was talking about was the research neuropathology report that is done on every case.
No, thank you very much, David. So really the takeaway message from this slide is that we aim to provide the best possible characterization of every single one of our donors to benefit research. And you see the additional resources that David talked about earlier, so we can go ahead to the next slide. And so important for, I think, our audience of researchers today, how do you access these resources, this information? And the way to do that is through our, or the Simons Foundation Autism Research Initiative online portal called SafariBase. In SafariBase, you are able to apply to access all of the data, all of the tissue. Once you have started an account, so you start like everything else online, you open an account. It is requested of you to provide some fundamental information, including IRB waiver or approval to be approved to view all of this information in SafariBase. Once you've been approved, again, you can see all of the clinical data, all of the tissue that's available. This is, of course, de-identified. I want to be clear about that. And you can then start to request the information and tissue or the tissue resources that are specifically of interest to you. And I know we're waiting for questions later, so we'll go ahead to the next slide. And that's it. We hope that you've, I guess, learned a lot through our presentation today. We're very interested to answer any questions that may have come up. And thank you again to Geraldine, Trish, Talia for having us here today. It's been a real privilege.
And I'll just say that that our website has an enormous amount of information. We've tried to keep this brief and not go into the weeds. But if you're interested in going into the weeds, you can do so on our website. I do think it's also interesting. Geraldine read a little note from one of our donor families, and we have a Memories of Hope section on our website where we have appreciations and remembrances of many donors. I'm humbled when I look at that page. And so both researchers who want to get more details about, how we process the brains and families to see, the whole process. That's a good source, for more information. But again, if there's specific questions that we don't get to today, feel free to email either of us, and we'll be happy to respond, to you. So let's go to the Q&A.
Wonderful. Dr. Amaral and Carolyn, thank you so much. Appreciate your presentation. And we've got a few questions that have come in the Q&A form. And I know that you talked about applying on SafariBase and then opening an account, and there are approved researchers. So we have a question that says, do qualified researchers include biotech companies, or should companies partner with an academic researcher to be able to access your collection?
Yeah, I'll take a stab at it, and then Carolyn can correct me. But the tissue is available both to biotech companies as well as to individual academic investigators. So either could, I think all of the individuals who go on for information have to be identified as a PI in order to then proceed. But the PI could come from academia or from the corporate world. It doesn't matter.
Great, thank you. Our next question is, how much clinical data do you capture from the brain donors with PMS? For example, disease severity across the different core symptoms, not only diagnosis and specific genetic change.
Mm-hmm. Yeah, it's an excellent question. And in particular, I'd like to respond to the disease severity. So this is one more reason why we use the autism diagnostic interview, because it takes incredibly qualitative information, behavioral information, and codifies it on a severity scale. And so that information is available to researchers for download. I am available to support researchers' interpretation of that codified data for whenever they would use it. Our other ways of capturing disease severity come through the interviews. We also do collect a significant amount of anecdotal information, but of course also medical records and educational records. Any information that a researcher is interested in, if they do not see it directly on Safari Base, they just have to ask us, and we'll be happy to do our best to provide it to them.
Okay, thank you. We have several more questions here. One is, do you require an IRB approval to access de-identified tissue samples, clinical data from your repository?
I think you just need to have an IRB exemption in order to be able to access the data.
Thank you. And next question is, how many years did it take you to collect the 7 PMS samples? Or in other words, how fast can we expect the PMS collection to grow?
So the information that we've shared with you today is regarding or specific to the donations that have been collected. As David mentioned earlier, this is our 10th anniversary. So it's taken us 10 years to collect the existing Phelan-McDermid donors. There may be, and I'll go back to look, actually, I think there are not. So we have also, as David mentioned, the ATP legacy tissue. There are not any Phelan-McDermid donors as part of that. We do hope also, as David indicated earlier, that through the sequencing, when we do get the results, that there may be some additional donors with Phelan-McDermid syndrome identified through that analysis. Other than that, again, we really rely on our community and partners for communicating about the importance of donation to help us grow our program.
I wanted to emphasize that, Trish, too, that what we've realized, so, in 10 years, you guys have built an organization as well. So, things change over time and you get better at things. What we realized was that we could have all the infrastructure in the world set up to process brain donations, but if the communities don't know about the potential for brain donation, it goes for naught, right? So this is why we're delighted to do these kinds of webinars with our partners in order to bring both the need and the potential for brain donation to various communities, and so it how long is it going to take until we get to 20? It's like the most difficult question that you could possibly answer, yeah, I guess, because it really depends on, as I was saying before, you hope it could be a long time. But in reality, there will be deaths in the Phelan-McDermid community, and we would hope that the families that experience that would consider making a donation to AutismBrainNet in order to foster your mission and your research. So all we can say is that we have the infrastructure established to accept any donation and as many donations as possible. And we just have to appeal to your families and other families to consider the possibility. And again, it's not a value judgment. If a family decides that they don't want to make a donation, that's perfectly fine. We completely understand. There's information on our website. I'm just reading a book by Sarah Gray. It's called A Life Immortal, about the donation of her newborn son who had anencephaly, and she follows her donation. And she talks about the fact that, for her it was really life affirming that she made the donation, and it spearheaded lots of different research. And the fact that it's all about communicating with the various communities. She actually worked with organ procurement organizations. And again, as Carolyn would say, if you have a sticker on your car, vehicle license or your driver's license, saying that you want to be an organ donor, that doesn't mean you're going to be a brain donor. It has to be a totally different process. So anyway, it's a difficult question, but we're rambling. I'm rambling now. So let's try and answer a few more.
Thank you. Thank you so much for that. We have a comment and a question. The comment is, thank you for your very interesting talk and extremely useful initiative. And we have a few questions. Can we see a list of all donations with details on the individuals? How would that work?
So once you log in, once you've been approved for an account in Safari Base, what you will immediately see is an inventory or a list of our donors' age, gender, diagnosis, medical, psychological conditions, cause of death, any genetic conditions that have been validated will also be readily available. And then after you see that initial sort of view, you can do a deeper dive into individual donors to learn more.
Thanks. And a couple of more questions from the same person. Is there a list of all publications, including research on post-mortem PMS effectiveness?
Yes, no, there's not. So we have investigators who've requested, for other conditions, 20 and 20, but we've had investigators that have received or are in the process of receiving more than 100 samples. So really, the investigator has to justify, based on the science that they want to do, how many samples are needed. And so what I didn't mention is that when an investigator applies for tissue, it goes to an independent science review committee to make sure that, number one, the investigator has asked appropriately for tissue. Again, we consider it important to ensure that we're providing tissue only to bona fide scientists and bona fide research. If an investigator requests tissue, but they've never had any experience in doing human neuroscience or human tissue processing, we then recommend that they request pilot tissue. And pilot tissue would be tissue not from our primary repository, but additional tissue that we've gathered to provide, basically practice tissue to investigators to make sure that they can do what they claim to do. If they do that and then are successful, then they can come back in for a request for the actual tissue. So the number really just depends on the science.
Okay.
I have a question. There's a question that is frequently asked to me, and I'd love to know your perspective, David. What is the possibility of doing RNA sequencing on brain tissue, post-mortem brain tissue samples?
Yeah, so we actually have investigators doing RNA sequencing, and actually not only of masses of tissue, but of single cells. So now it's gotten so sophisticated that people are looking at the transcriptome either on tissue that has been maintained so that you can actually see where the RNA is expressed in different portions of the different cell types of the brain. And others are taking nuclei and looking at single nuclei in different regions of the brain. We have a very big project going on in the hypothalamus now, where people are looking at single nuclei in different portions of the hypothalamus and doing RNA sequencing. So, or RNA and transcription. So I think it's entirely possible.
Thank you.
And we have one more question here, talking about the two different methods that you were discussing earlier with the brain. And for the families, could you explain a little bit more on why you do two different methods?
Sure. And I won't go too much into the weeds, but it turns out for doing the kind of study that Geraldine just actually asked about, you can't. So when we divide the brain, the frozen brain is just cut into coronal slabs. You can imagine it like a loaf of bread cut into slices through bread and then frozen. We do that in order to freeze it most rapidly and keep the tissue in the best possible quality. That tissue is optimal for doing analysis of DNA, analysis of doing RNA, of looking at how RNA is expressed in different regions. If you wanted to do genetic studies, you would want to use that tissue. But other people are looking at pathological signatures in the brain, or counting neurons, or counting glial cells, or looking for immune responses using antibodies, for example. The frozen tissue is not very good for that. So what we do is the other half of the brain, we actually fix it in a solution of paraformaldehyde. And that fixes it in place so that all the proteins are fixed in place. And we do it in a way that allows the tissue not to be changed very much. In the old days, people would just take a portion of the brain and put it into a container with formaldehyde and leave it go for weeks or months or years. The problem with that is that it changed the brain so much that you couldn't do, a lot of science with it. So we fix it just minimally, only 36 to 48 hours. And then we cut it. We take cut slabs that we have and cryoprotect them. So we make it so that when you freeze it, all the tissue is going to stay undistorted, and then we put it into the freezer, and then we can take a piece of that tissue out, cut it into sections, and microscopically it looks like tissue as it was in the living brain. And you can count neurons, you can count glial cells, you can do all kinds of studies. You can look at transmitter substances in the brain. You can do in things like in situ hybridization and others. So there's different science that you can do on one side of the brain versus the other. And we one of the hardest jobs for me and for my scientific colleagues is to predict what science is going to be being done 10 years from now or 20 years from now. And what we have to do is make our best possible guess as to how to process the tissue in order to enable that science that's going to be taking place over the next decades. And by doing the half fixed, half frozen, we think we've come up with that kind of a solution.
Thank you. And thank you for answering all the questions, and thank you to everyone. I think we're out of time now, but we appreciate you posting questions in the question and answer section. If you have further questions, we've put up in the chat the email address for requests at autismbrainnet.org for more information. We've got the websites there for for you. And if we did not answer all of the questions at the Q&A, we'll take a look at those as well. And we would just like to thank our distinguished speakers for talking today and providing information today. We really appreciate it. And if you'd like more information, please reach out and either on the website or at request@autismbrainnet.org. And next slide, please. And we'd just like to let you know that that this Resources for Researchers webinar series is brought to you by the Cure-Shank Biomarkers and Outcome Measures Consortium. And if you'd like more information on the consortium, you can visit our website, or you can contact me at consortium@cureshank.org. And thank you again, Dr. Amaral, and thank you, Carolyn, for being with us today.
Our pleasure. Thanks so much for the invitation.
Thank you.
Thank
Take
y'all
care.
very much.
Bye-bye.
Okay, bye, everybody.